It looks like we're ready to go for this final plenary session. We have 3 speakers today, and we'll start with Claude Desplan from, or I should say Claude Desplan, from New York University, who will be talking about the generation of neural diversity. Claude, it's yours.
Okay, good afternoon for all of you. And for me, it's a first for such a large audience by Zoom, but I will try to do what I can. Okay, so I'm a developmental neurobiologist, and my major interest is to figure out how we can generate the huge diversity that you have in our brains. And so if you look at the human brain, it's immensely complex with about 80 billion neurons, maybe 100 billion neurons, and each one of them makes about 100,000 connections to each other, and this indicates that there is a large diversity. On top of that, there is a very large number of diverse neurons, which we don't exactly know the amount. We don't know how many neural types there are. And so it's a bit too difficult for me to address, and therefore we chose to have a system which is a lot more simple, which is a fly optic lobe, which is actually a fairly still complex structure, 60,000 neurons, which means about 1 million times smaller than the human brain. And yet, you know, the fly can still perform very acrobatic flying and can escape you when you try to catch them. Therefore, it can perform very well. The whole brain of the fly is about 100,000 neurons, which means most of the neurons in the brain are actually part of the optic lobe. The optic lobe are made about 200 neuron types, and most of them have been characterized starting 100 years ago with Cajon and Cajal. And recently, Janelia Farm has actually managed to establish the connectome of most of those neurons, which means you know which neuron exists based mostly on morphology, many of them on function, and we know how each one of those neurons is connected to the other neurons. Again, in a very highly performing system. So what I'm going to tell you today is that what we have done in terms of using single-cell sequencing to be able to address this question. And you're going to say, oh no, yet another talk that talks about single-cell sequencing. But you will see, I think this technique has been invented for us because it's such an amazing system to be able to address the question we try to ask. Okay, so again, we know quite a bit about the neuron, and here it's going back like almost 40 years ago when Fishbach actually defined most of the neurons based on morphology, and you can see still a very complex structure, and I'm going to try to describe it to you and to see how we can understand how it functions. So it's actually a picture from Robbie Singer's lab, which beautifully describes how the system is organized. So what you have in the visual system, the eye, okay, and there is about 800 unit eyes, which are basically detecting 800 pixels, very low resolution, but still very highly performing eyes. And of course, the eye, the photoreceptor will project to the first layer of the optic lobe called the lamina. And it's going to do so is what we call a retinotopic organization, which means things which are here project here, things which are here are going to be here. And it's true for every visual system. In every system, you maintain the image of the world on each part of the brain when this project. So you might have 800 cartridges in the lamina, and the lamina has for main purpose to detect motion. Then, and you can see here how those photoreceptors are projecting into the lamina. These are the photoreceptors that are involved in motion detection. Other photoreceptors, which are most involved in color vision, are projecting into the medulla part of the optic lobe, again, with 800 colors, again, maintaining the retinotopy. And then neurons from the lamina project to the medulla to convey further importance of the further processing of the motion system. And then it goes to higher processing center until it goes to central brain, where the flies will make a decision to take action in response to the visual stimulus that it got. Okay? So this is actually a fairly complex structure, but still manageable. But 20 cell types in the eye, mostly photoreceptor and support cells. Then the lamina is a very simple structure with just 5 cell types. And those cell types are just involved in the first process of motion vision. And the medulla is really the most complex structure where it's about 100 cell types are processing almost everything, motion, color, edge detection, everything. Okay? And then to higher processing centers, the lobula, which I will not talk too much about, it has about 60 cell types. And then you go to the central brain, which is a huge complex structure with many, many, many different cell types. Okay, so what kind of cell type do we find in this system? So we can actually define them here by their morphology. And what you have here is basically a close-up of this part of the medulla, for example. And you have one neuron which is in the blue region here, which is mostly the cell bodies. They're going to project into the red region and somehow connect to photoreceptor, which are the green part here. So if you take one single neuron, you can see how it's going to be projecting. So what you have to realize is because you have 800 ommatidia, 800 cartridge, 800 column, what you have, in fact, 800 circuits running in parallel to one another, okay? Which means these 100 cell types that you see here, they are basically present around each one of those columns here. And basically, if you can understand the circuit into one of those columns here, you understand the whole thing because you just multiply everything by 800. So we want to do that. We want to understand what's going on there. And again, you have to realize that we know quite a bit about the system because we know most of the connection from EM for this part of the brain of the fly. So what kind of neurons are contained here? So we can take an example of a neuron that will have his cell body here in the blue region, and then it's going to project to talk to the photoreceptor, and then going to project and to go to higher processing center. This is one example here. And you can see in red as a photoreceptor, the end of the photoreceptor here, basically the end of green stuff here. And you can see the neuron is a monopolar neuron, which means there's a one long process that actually sends dendritic arborization, postsynaptic arborization to collect information from R8, from R7. Here, there is some processing of information, and they go out of focus to higher processing center. We call this a unicolumnar neuron because we imagine there is one of each for each one of the photoreceptor, which means you expect to get something like that, which means you expect to get about 800 of these neurons, always the same, one on each of the columns. But then you have neurons that compare the different pixels. Basically, they have a larger receptor field that contacts multiple columns here. So they're called multicolumnar neurons. And in the tail, you expect to get quite a lot fewer of those neurons to cover the receptor field of the fly. So something like 75 neurons. So if you do that over and over again, you can define a large number of neurons, again, about 200 in the whole optic lobe, 100 in the medulla, and they come in different flavors. You have first local neurons, which mean neurons that remain in the context of the medulla, as compared to projection neurons that collect information from the medulla and then go to higher processing center. But more importantly, what I want to tell you today, you have two types of neurons that I just described to you, those unicolumnar neurons, either projection or local, which basically has one per column. So which means you have about 800 each, and you have about 30 cell types of this type. And then you have the multicolumnar neurons, which are fewer. There is fewer for each of the cell type, but there are many more cell types. There are about 70 cell types which belong to these multicolumnar neurons. And those are going to be important for processing information, comparing different pixels in the visual field. So what can we do to be able to take advantage of this morphology and to try to get into the guts of those neurons and try to understand how they function, how they are made, how they make their projections, how they connect to each other, and understand how they do. Of course, to do that, we need to be able to have access to their transcriptome to understand which genes they express, which cell addition molecules they express to be able to contact to each other and to make connections. And so we started a few years ago taking advantage of the fact that actually there is so much which is known about those neurons, and in particular we and others, and in particular Genelia, have defined a large number of drivers, which means molecules insertion into the genome that can drive GFP expression specifically in one neuron cell type. For example, in that neuron, in that neuron, in that neuron. And so what you can do, you can take a flies which express this GFP in one neuron, for example, in TM1, and purify by fax all the TM1 neurons, and then do deep sequencing, bulk sequencing over these neurons. We did that for 21 neurons that way. It was very hard, and faxing each one of them, it was difficult, but we managed to do that. And then recently, Genelia published 51 of those neurons. And so we now have a collection of 50 neurons. It's actually 50 neurons out of the whole optic lobe, which means 50 out of 200 neurons that exist. And so we're going to continue and take advantage of that. But then, of course, about 2-3 years ago, single-cell sequencing came out on the market, and we decided that it was a perfect technique for us to use and to be able to take advantage of that, not to get to one neuron one at a time, but to get to all the neurons all at the same time. And so what you have here is actually a pretty amazing picture, which was obtained when we sequenced about 110,000 neurons, cells, okay? And we can cluster very, very well. That means a very precise cluster. I'm sure you have seen a lot of single-cell sequencing result, and usually it's a big blob with different colors. Here you can see each cluster is very different because each one corresponds to a very specific cell types. We can actually identify about 169 neurons and some glia. And of course, because we have the bulk sequencing of many of those neurons, and because we have done a lot of work on some of those neurons, we could identify about 65 of those clusters, put a name on those clusters to identify what they are, which means we know who they are. Because the problem with single-cell sequencing is that you get the transcriptome of a neuron, but you don't really know what this neuron is unless you have some markers that allow you to identify it. And so you remember we had two classes of neurons that we had in the optic lobe. So neurons which are unicolumnar, which are 800 neurons per brain, which means a large number, and the neurons which are multicolumnar, which are fewer neurons per brain. And you can see that is exactly what you see here, because all these big clusters, which are quite abundant, correspond to all the neurons which are unicolumnar neurons, which are present at 800 neurons per brain, whereby it's a smaller cluster, which are a bit more difficult to identify because there are fewer cells, correspond to multicolumnar neurons, and those come in flavor between 5 neurons per brain to all the way to 800 neurons per brain, and anything in between. And so now what we have a description of a large number of neurons, and if you realize what happened there, it's quite critical, because what we have, we know how all those neurons, which genes they express. And on top of that, as I told you earlier, we also know that connector, which means that we know how this neuron connects to that neuron. We know how many synapses it makes, and we can really figure out which cell adhesion molecule it has, which neurotransmitter it has, and try to figure out exactly how the brain is being constructed. So we hope that this data set will allow us in the future to be able to understand how basically the brain is constructed. One of the problems, however, is that this actually is the transcriptome of the adult neuron. And of course, connections don't occur in adults. They occur much earlier during development. And therefore, it's actually a view of how the neurons function rather than how the neurons were made and how they connected to one another. Which means this is very useful, but it's only detecting how the neurons function, adult neurons. Which means if we want to understand how the neurons connect to one another, we need to be able to look earlier in development at the time when they make the connections. And I'm going to give an example of that to illustrate what I'm just trying to say. This example actually is this big, huge blue cluster here. And usually if you do single-cell sequencing and you see a large cluster like that, you know it's crap. Some crap neurons, some or the ambient RNA. In this case, it's not actually crap, because actually these neurons here, the T4A, B, C, and D, and T5A, B, C, and D neurons, are likely the best studied neuron in the fly brain, because that's the first neuron which respond to motion. And yet, although they have 8 neurons which respond differently to motion, they all seem to be part of one single cluster in the adult. And you think about it, it might make some sense, because those neurons might actually function the same way in the adult. But since they have different connections to be able to detect motion, they are going to be different earlier in development, but we don't see it here. So we need to go back earlier in development. But first, let me tell you what those neurons are and how they can differ from one another. Okay? So those are the first motion-sensitive neuron in the brain. And those motion-sensitive neurons are called T4 and T5. And there's a cell body here. You remember, this is the retina, the lamina, the medulla, the lobula, and the lobular plate. And those 4 neurons, you have two types, the T4 and the T5, and they simply differ by virtue of the fact that T4 detects the motion of a bright edge on a dark background, whereby T5 detects a dark edge on a bright background. And actually, every visual system that detects motion has always this kind of on-off pathway to be able to distinguish between the two types of motion. So if an object that comes in the sky, for example, a bird coming to eat flies will start with a dark edge, followed by a bright edge when it goes back. Okay, so there are 2 types of neurons which are actually projecting to very different part of the optic lobe, because T4 projects to the depths of the medulla, whereby T5 projects to the surface of the lobula here, okay? So very different projections, but I told you there is actually 8 neurons. Where does 8 come from? Simply because there are 4 types of T4 and 4 types of T5. One T4 actually is a neuron that actually collect information from the depth of the medulla and project only to the first layer of the lobular plate. And these neurons are always involved in front to back motion. When you fly at high speed, the world basically goes front to back, and therefore those neurons are going to be important for being able to detecting that. And same thing for T5, they also project here, but now they go to the first layer of the lobular plate. In the second layer, you have the neuron the type B, which goes back to front. So of course, most flies don't back up, but when you rotate the flies, the right eye is going to see front to back, the one where the left eye right see back to front. Okay? And flies in 3 dimensions, therefore there are also neurons that project to the third layer of the lobular plate that see up motion, whereby the fourth layer is down motion. So what you have here, 8 neurons are clearly connected very differently, either to the lobula or to the medulla, and to different layers of the lobular plate, and yet in the adult, they form exact same cluster. That means they have exact same transcriptome, or nothing we can distinguish very clearly. Okay? So we said, let's try to look back in development and see what happens earlier in development, for example, around 50% pupation, when most of the connections are being made. But of course, at 50% pupation, there is no way you can identify the neuron because they have no form of shape. They have not yet acquired their shape, and therefore you don't know who they are. You cannot recognize them by morphology. But we say, what if we go to look at from our data set in adult and try to go back in earlier and earlier and try to match this time point, for example, with the adult time point, with a premise that actually at this point here, the neuron expresses a transcriptome which will resemble the transcriptome of the adult, and then we can jump back and forth and forth and forth. Okay, so we can go back from adult to 70, 70 to 50, 50 to 40, 30, and 20. And hopefully we should be able to assign an identity to all of the cluster we might be able to identify during development. And exactly what we see. In fact, this works beautifully well. So a postdoc in a lab, Neshat Ozel, developed a software to be able to try to compare those two sets to do identification transfer, which means you cluster the neuron at P70, and you take advantage of this huge data set here, and you can match perfectly each one of the clusters to one cluster in the adult, which means you can easily find a name to all of the clusters that here, match them to the cluster in the adult. And then you can do the same thing from 70 to 50, to 50 to 40, 40 to 30, all the way to P15. So even at P15, when they were just born, and you can see actually some of them are still being born at this stage, you can actually match all of the clusters. It's almost perfect. So everything matched very well. And now we can say, so what happened to this T45 cluster at, for example, 50% pupation when those cells were actually making the connections? And actually, it's exactly what you expect, because now what you can see, 8 beautiful clusters, of course related in the T-SNP plot, because indeed those cells are related, but they're clearly distinct between the front to back, back to front pathway, or the up and down pathway here, which are separated, and you can see the T4 and the T5. So that means that indeed at the time when those neurons are making that connection, you can easily distinguish them, and therefore you can actually identify the cell adhesion molecule that allows them to further connection, or the transcription factors that control that. So what you have here, you can see the differentially expressed gene, and clearly you can see that you have the front to back and back to front different from the up and down, and you can see also that the front to back is different from the back to front, and on pathway versus off pathway. And if you look at the type of molecule which are, so these distinctions actually correspond to the developmental distinction between those different cells, because they develop from 2 precursors that make 2 successive asymmetric cell division, which are not controlled. And basically, they first distinguish the 2 precursors between the horizontal system, front to back, back to front versus up and down. And then they distinguish between front to back versus back to front, and then distinguish on and off pathway, and same thing here. And you can see this reflects completely the developmental pathway in terms of the differential gene expressed. And if you look at which gene expressed there, these are actually cell adhesion molecules, which for the most part are part of large families of cell adhesion molecules, which allow, presumably, those cells to make a specific connection. For example, the bits are cell adhesion molecules that can connect to sides, and the DPR and DIPs are just cell adhesion molecules that can contact to each other. Now, I don't want to give the impression that it's going to be very simple, because as we know from all the 30 or 40 years of developmental biology in neurobiology in flies, we know that there is no single gene that can really control the connection between two neurons. There are a few examples and some of one we published recently, but overall it takes a combination of those different cell adhesion molecules to be able to allow two cells to connect to one another. And we are very busy trying to figure out what's going on there and what happened there, okay? Okay, so, so this is at 50% pupation, where you can see you have these 8 beautiful clusters. And what can we learn from this? Because what can we learn from this development? So I circle in red and in blue here 2 other neurons, which actually are interesting neurons because those neurons also, like for T45, were only generating one type of neuron in the adult, one cluster in the adult. And yet at 50% pupation, they form 2 clusters in this case and in that case. Let me give you the example of DM3. DM3 are large neurons whose function we don't know very well. We believe they're involved in motion vision. In fact, they are 1 cluster in the adult, 2 clusters in development. And we realize actually that actually there are indeed 2 types of DM3 neurons, those that project in one orientation and those that project 90-degree orientation. Okay, a bit like what we saw with the T4 and T5 neurons. They were projecting front to back, back to front, or up and down, and again 90-degree. And again, if you see what distinguishes those different neurons, they're quite different. They express different cell adhesion molecules at 50% pupation, which again are part of family of cell adhesion molecule and also transcription factor that control how they're going to, in combination, as they control the different orientation of the dendrite, which are projecting, where those neurons are projecting. And we have actually quite a number of those type of distinction we can see. And I don't want to give the list. There is so much we have to dig in to be able to understand what's going on there. I want to give you just one more example, which is neurons which doesn't exist in the adult, but exist at all the developmental point during pupation, during development. There are 2 clusters of neurons. And those neurons are of interest because they disappear in the adult. We cannot find a match. And it's not that actually it's a mistake or a problem in the software. No, in fact, these neurons are a very large family of neurons which actually are surrounding from the dorsal or the ventral part of the optic lobe. They never enter, they just flank the optic lobe, and they are present during development, but then they disappear in adulthood. So here they are. You can see those neurons, one ventral patch, one dorsal patch, okay? And later on, at 85% pupation, which is just before they close, they start expressing genes which are involved in cell death, in apoptosis. And you can see caspase standing here. And then in the adult, they're gone. We don't see them. If you prevent cell death by P35, which is a molecule that prevents the caspase from acting, what you can see, those neurons survive, and you can see they're still there. So we're very interested in figuring out what's going on there. And for those who know mouse development, this, to some extent, resemble what the chiral reticulus cells in the cortex of mammals that seem to be there and they disappear in adulthood. So you can see a lot of information that we have here, and we are digging very deeply to be able to understand really how basically transcription pathways and cell adhesion molecule are able to control the formation of those neurons and the different shapes. So what I told you is that we know quite a bit now about how these many cell types that you have in the optic lobe, in particular those that you have in the medulla, are acquiring their the specificity in terms of making the specific connection. But the question that remains is how are those diversity of neurons generated? How is it generated? How do you create 100 cell types for making the medulla, for example? And so I don't have time to go through a lot of data, but what you have to realize actually, if you focus on the medulla, is that the medulla comes actually from a very simple monolayer monoepithelium. And this crescent-shaped monoepithelium at the outer edge will start producing neuroblasts, which means the neuroepithelium cell will become neuroblast. And as time goes, the whole neuroepithelium will be transformed into a neuroblast over a period of 3 days. Those things are a bit like that. So you can see with time, what you're going to have is that the neuroepithelium will be transformed into neuroblasts, which are going to be formed over a period of 3 days. So that means you have all neuroblasts here and young neuroblasts here. And this, something very striking about that, actually we counted about, and this number is not very precise, about 800 neuroblasts. And this actually suggested that maybe what happened is actually in the formation of this medulla structure, which remember has 800 columns, you have 800 neuroblasts, which suggests that there is about each neuroblast can produce all the neurons for each one of the columns. It's not absolutely true, but it's kind of true. In fact, it's true that actually each neuroblast produces a large number of the neurons that surround each one of the columns. And you're going to see exactly how this happens. Okay? So if you make a cross-section and you look at 90 degrees, again, the neuroepithelium is here. And you can see this neuroepithelium will become a neuroblast. And neuroblasts do what stem cells do. They divide asymmetrically to give rise to GMC, an intermediate precursor cell. They divide once more to give rise to 2 neurons. And then an hour or two later, the next epithelial cell will become a neuroblast and will do the same thing. But this first neuroblast will keep dividing. And it's going to happen something like that when at some point you're going to have young neuroblasts that just forming young neurons, and then old neuroblasts that actually form young neurons still, but also have formed old neurons which are here. And therefore, what I argue is that this column of neurons here come from one neuroblast, form most of the neurons that surround each one of the columns. How do they do that? How can one neuroblast produce 100 neurons, 100 different types of neurons? And actually, the solution was provided many years ago by Chris Doe when he actually told us that the neuroblasts in the ventral nerve cord of the flies could go through what they call temporal series of transcription factors. And those transcription factors means actually the neuroblasts start expressing a transcription factor, then switch to another one, another one, another one, another one, same neuroblast, and every time it produces different types of neurons. We found the same thing here, except the transcription factors that we find are totally different from those of Chris. But what you can see here is actually the neuroblast that is born express homothorax, then clufus, and homeobrain, islets, lobiped, dactyteles, and every time it produces one neuron and basically able to produce a different type of neuron. But of course, here I give you a series which we find a bit by chance, a 6 or 7 transcription factor, and this might not explain how you can generate 100 neurons from one type of neuroblast. And so we decided to try to figure out how this happened. And again, the way we defined those temporal transcription factors was a bit because we had available antibodies or we had some educated guess, but it was in no way systematic. So we said, can we use a systematic approach to be able to identify the different transcription factors that can indeed specify this large variety of neurons that you can find in the optic lobe? And so again, so what we did, we actually tried to go back now at the time when the neurons were being produced, when the neuroblasts were working, and try to understand how those neuroblasts could actually go through many type of temporal series and temporal windows to produce all the neurons which are required for that. So if you look at during the larval stage, when the neurons are being produced, it's a bit of a mess because at this stage, the neurons are just starting to form, and most of the clusters that you see actually are very elongated clusters because they're just neurons in a process of developing. So they're not all in the same shape in terms of transcriptome because they're being produced over a period of 3 days, and therefore they're all very elongated. I don't want to talk about that too much. So neurons are actually here on the left side, and I want to focus my attention either on the GMC, but mostly on the neuroblasts. The neuroblasts are the neuroblasts which are here, and that's the neuroblasts that produce the medulla. They're going to be neuroblasts that produce the lamina, and these are going to be neuroblasts that produce other structure. Okay, so I want to focus my attention on those neuroblasts here that produce the medulla. And the question we can ask is, can we use ways to bioinformatically define which transcription factors are going to express with time?
Claude?
Yes? Yes?
You have 1 minute.
Okay, so I'm almost done. So in fact, what you can see is that you can use program like a monocle, which is a program which calculates pseudo time, which means it compares different transcriptome of different neuroblasts, and actually is going to see how they can be ranked in order of pseudo time, which means how a given neuroblast can evolve from one state to another one. So if you look at now at what happens here, what you can see is that you can actually, again, looks like an in situ. In fact, it's just a bioinformatic representation of the neuron. That the temporal series I told you before, homothorax, islets, lobiped, dactyteles, is reflected by the pseudo times that you have in the single cell sequencing here. Okay, and you can see here, I can note all the staining. So now what we have, we actually can see that we have all the known transcription factor, but what's happened in between? Can we identify more transcription factor which are in between? And the answer is yes. So in green, what you have, the known transcription factor, which identify homothorax, islets, lobiped, dactyteles. And in between, we've identified a number of new temporal transcription factors which appear to be expressed up and down during a very short temporal period to be able to produce more neurons. And we have validated most of those neurons here. So in conclusion, what I just want to tell you is that, again, these techniques of single-cell sequencing, and which actually are really embraced, and it took me a long time to become a genomicist, if I can call me like that. But I think I realized that how powerful this technology is, especially for us when we have this large variety of neurons. And the goal here will be that by looking during development at the transcriptome of developing neurons, we will be able to understand how those connections that we have in a connectome can be established and how we can understand basically the rules of the connection. Again, very simple rules will be much more complicated and most likely add activity-dependent patterning in vertebrate, but we will be able to define the rule of connection of transcription factor. Okay? So I want to conclude here and give credit to people who have done the work. This work was pioneered by Nikos Konstantinides with the help of Katarina, and then was taken over by PhD student Neshet and Felix, who have done an amazing job looking at this pathway I described to you. And of course, I can tell you everything else we do in the lab, and I've highlighted in yellow the PhD students who are contributing to the lab, including Felix. Thank you very much.
Thank you.
And I'm happy to answer any question with the chat room.
So for those of you who have questions, you see that there is a question and answer button on your screen, and you're welcome to send questions, and Claude will be happy to answer these. So now we move on to the next speaker, who's Sarah Otto from the University of British Columbia. And she'll be talking about selective interference and the evolution of sex. Sarah, it's yours.
Excellent. Thank you very much for this opportunity to speak to such an amazing virtual audience. So I'm going to talk today about selective interference and the evolution of sex. Hold on a second. There we go. And I'm going to tell this story in 3 acts, starting with the history of thinking about selective interference. And before I get started, I want to discuss one of the key players in this story, and that's linkage disequilibrium. Linkage disequilibrium is just a measure of association or correlation. Technically, it's a covariance between the alleles carried at different loci. The way I will be defining the sign of linkage disequilibrium is that it's positive if the beneficial alleles tend to group together and are found more often on the same chromosome, as well as the deleterious alleles. If those are overabundant, then there's positive disequilibrium. By contrast, the disequilibrium is negative if beneficial alleles tend to find themselves in genetic backgrounds with deleterious alleles at linked sites. So the first mention of the topic of selective interference was in the 1930s by Fisher and Muller. Here's what Fisher wrote. For unless advantageous mutations occur so seldom that each has had time to become predominant before the next appear, they can only come to be simultaneously in the same gamete by means of recombination. And Muller wrote something similar. Without sexual reproduction, the various favorable mutations that occur must simply compete with each other. Mathematically, Crow and Kimura were the first to explore this question in 1965. Alongside their mathematical treatment, they also provided these so-called Muller diagrams. Along the x-axis is time, and along the y-axis is just an array of the genotypes within a population. And they noted that in a small population, mutations will appear and rise to fixation pretty much independently from one another over time. It's in a large population that we see selective interference because mutations can arise nearly simultaneously in very large populations, but without recombination, they will arise and potentially increase in frequency for a while, but eventually they will disappear. And so what Crow and Kimura asked was, how long does it take for this kind of nesting time from the appearance of one mutation destined to fix and the next mutation destined to fix in the same lineage in an asexual population? And they compared that to the rate of adaptation in the sexual population. What they showed mathematically was that sexually recombining populations could adapt faster, and actually a lot faster, especially if the population was large and mutations were weakly beneficial. because then there was a lot of mutations that would happen simultaneously and they would take a while to resolve. And so there was more interference and more mutations lost in an asexual large population. But soon thereafter, Maynard Smith in 1968 raised a counter argument and developed a mathematical model that questioned whether selective interference was important at all. In this model, he started the population at mutation selection balance prior to an environmental change, and he assumed that there was no fitness interactions or no epistasis between the loci, so that their fitness effects just multiplied together as shown here. He then demonstrated that if there's random associations, that is no linkage disequilibrium to begin with, that there never would be linkage disequilibrium during the spread of the beneficial alleles. D is 0 to start and it stays 0 with multiplicative interactions between the loci. And if the alleles are already randomized on the chromosomes within a population, recombination is going to make no difference to the speed of adaptation in this case. Maynard Smith thought that the main difference between his treatment and Crow and Kimura's is that he was treating mutations not as unique events like Crow and Kimura did, but as recurrent events, and explored this question at mutation selection balance. But there were hints already in the literature that there was more going on. Hill and Robertson in 1966 published this paper exploring the limits to selection. And they were particularly interested in breeding programs and how different breeding programs would affect the rate of response to selection. They used simulations to show that even if a population is in linkage equilibrium with many alleles present, just like Maynard Smith had, the advance under selection is reduced when genes are tightly linked, even if there are no fitness interactions between loci, like Maynard Smith assumed with multiplicative selection. Here's an example graph from their figure. The strength of selection at the other locus, at a second locus, is shown here on the bottom, and the fixation probability of a beneficial allele at a focal locus is shown here on the y-axis. The tighter the linkage, NC is the number of recombination events per generation, the tighter the linkage, the stronger the interference, and the lower the fixation probability of this beneficial allele was. Over all of their simulations, they showed that selection at one locus always interfered or had a negligible effect sometimes, like in these cases with weak selection, reducing the fixation probability of beneficial alleles. So which is it? At this point, Felsenstein in 1974 published a really important synthesis paper that took a look at those studies that had found an advantage to sex and recombination when there were no fitness interactions between loci, and those papers that did not find such an effect. And he noticed a major difference, and that was how they modeled evolution. In the papers and treatises that did find an advantage to sex, they tended to include stochastic factors, chance factors, including drift that would happen in a finite population. Whereas those models that don't find an advantage to sex, like the Maynard Smith one, were deterministic in nature and assumed infinitely large populations. Well, this is still a puzzle. Okay, that's interesting, but it's somewhat puzzling because one normally thinks of drift and stochasticity as just adding a little bit of noise to a system, not fundamentally altering the direction in which evolution occurs. And yet here we're seeing an advantage to sex in stochastic treatments that we don't see in deterministic treatments. Well, let's dig in a little bit further. Hill and Robertson pointed out that the dynamics of the linkage disequilibrium obey this type of equation, where the disequilibrium in the next generation is equal to a function of the disequilibrium in the previous generation. K1 and K2 are just constants that depend on the selection and recombination in the system. So this shows that if there's no linkage disequilibrium to start with, then it will stay 0, just like Maynard Smith pointed out. But if there's variation in disequilibrium, this is a nonlinear function. So we're not going to see the same effect in the next generation of variation in disequilibrium. And indeed, we find that if there's no disequilibrium to start with, then in the next generation, the expected amount of disequilibrium is negative. This is the way I like to think about it. When, by chance, beneficial alleles are found on the same chromosome, that's positive linkage disequilibrium, selection becomes more efficient. There's plus and plus combinations, good alleles on good genetic backgrounds. And that makes selection very efficient, leading to the rapid rise in frequency of the favored chromosome, the rapid disappearance of the combination with only deleterious mutations. And this in turn causes linkage disequilibrium to decay rapidly. And you can see that in this equation. In the case where by chance there's positive disequilibria, there's a higher mean fitness, a higher denominator. That reduces this equation and reduces the net effect on disequilibrium in the next generation. By contrast, if beneficial alleles occur by chance on poor genetic backgrounds, there's negative linkage disequilibria created by chance, then selection slows down. The genetic variation's kind of hidden in chromosomes that carry good alleles alongside bad alleles on the same chromosome. And this slowdown of evolutionary response freezes the population in a state of negative disequilibria. In this case, the denominator is smaller, the mean fitness is lower, and it causes this whole fraction to be a larger quantity. The disequilibrium becomes negative. So the end of all of that is in the presence of stochastic forces, drift, as well as selection. The chromosomes that we'll see after a period of evolutionary time have accumulated an abundance of good alleles on bad genetic backgrounds. A lot of genetic variation becomes hidden, and that limits the response to selection. So the way I see this is selective interference is a broad umbrella term that encompasses a lot of different phenomena that we've been talking about in evolutionary biology, including Hill-Robertson effects, Fisher-Muller effects, clonal interference, and genetic draft. Felsenstein suggested we use the umbrella term Hill-Robertson effects for all of these, but I suggest that we use instead selective interference, simply because I think that's a more accessible term for people outside of the population genetics literature. But I agree with him that Hill and Robertson were clued into the important effects going on here. I would also like to say that even that Muller's ratchet belongs under this umbrella too. Muller's ratchet is a phenomenon where if the most fit combination is lost from a population by chance in any generation, then it cannot be recreated in the absence of sex and recombination. And so there's a loss of that best class, the moving over the distribution, and that can ratchet and happen again and again. But in terms of the selective interference discussion that I've been having, what this amounts to is the loss of the best class and an overabundance of good alleles on bad genetic background, just like the other examples of selective interference. So again, drift in the presence of selection tends to lose the best class, or the best class becomes less abundant. The other important point of this large umbrella term of selective interference is, again, it doesn't really depend on the nature of the stochasticity. It doesn't depend so much on whether it's random genetic drift, the sampling of offspring from the parental generation, or if it's the chance background on which mutations arise. Either source of variation is what's needed in order to see this phenomenon. And it doesn't depend on the uniqueness of mutations. Given the complexity of the simulations requiring tracking multiple alleles at multiple loci, Hill and Robertson concluded that it seemed unlikely that any general theory could be constructed. But since then, we've had some analytical success. My colleagues and I have been using a variety of approaches, including using branching processes to see how much selective interference reduces the chance of fixation of beneficial mutations, as well as tracking moments to show how strong selective interference becomes and how it reduces the rate of adaptation. I want to take a moment now to ask what evidence is there for selective interference of the type I've been talking about. The most common such study has looked at whether or not sexual populations adapt faster than asexual populations. And this has been demonstrated in a number of laboratory experiments in a number of organisms, as shown in this experiment from Goddard et al., whereas the sexual yeast populations adapted faster than the asexual ones. One of the challenges of these studies, though, is that it's difficult to tease apart the mechanism. So sex is adapting faster, but is that due to selective interference? Or maybe there's epistasis going on, that is fitness interactions, not the multiplicative independent assumption that I've assumed so far. Or there might even be direct pleiotropic effects of sex. For example, epigenetic reprogramming following meiosis can be quite a bit different than that following mitosis. We get a little bit more insight when we can peer inside the genome and look at what's happening alongside these experimental evolution studies. A very nice paper from McDonald et al. In yeast was the first to do that, to compare sex and asex and the signals that they leave inside the genome. So what they found was that were classic signatures of selective interference. The beneficial mutations that fixed were ones that were nested within lineages that carried other beneficial mutations. So we see these cohorts of beneficial mutations in gold fixing together with one another. Whereas in an asexual population, that is, in a sexual population, it's entirely different. And the beneficial mutations appear to arise and spread to fixation pretty independently of one another. Furthermore, when they looked at the deleterious mutations in blue, they often found that these cohorts of mutations spreading to fixation often carried other things that were nested within the same genetic background, carrying deleterious mutations to fixation. That didn't happen in the sexual populations, however. There were rounds of sex, and when the rounds of sex happened, they tended to uncouple deleterious mutations from the genetic backgrounds that they were on, and then you'd see the deleterious mutations going away. And so in the end, the sexuals were adapting faster in large part because more mutations could rise and fix independent of the background in which they appeared, and as well, the deleterious mutations didn't get dragged along with them. How about out in the field? Well, we don't have as much data about organisms in nature. Neiman and al. Recently reviewed field-based studies and pointed out this lack of testing of whether sexuals or asexuals adapt faster. They did point out, though, that there had been studies looking at the accumulation of deleterious mutations in sexual versus asexual lineages. And in the majority of cases, asexuals accumulated deleterious mutations more often. This is consistent with selective interference, but as I mentioned, it's impossible to be sure that that's the mechanism as opposed to other features that have to do with pleiotropic effects of sex or epistasis. And one other type of example that has looked at whether selective interference is important has examined regions of the genome and counted up the substitutions that occur, that are non-synonymous and that are synonymous, and how this depends on the recombination rate in that region of the genome. Using this method, Castellano et al. Showed that the rate of adaptation in Drosophila melanogaster is reduced by 27% on average because of selective interference at neighboring sites, and that's averaged over the whole genome. When they looked instead at genes that were within high gene density regions, and that had high mutation rates, they found a whopping 60% slower rate of adaptation because of this selective interference due to neighboring loci. So far I've been talking about the impact that selective interference has on adaptation and whether or not it slows adaptation in sexual versus asexual populations. And that suggests that I'm talking about a group selection level advantage. But I want to shift in this second act to talk about how the evolution of sex and recombination can occur within a single population. And to describe how that's possible, I need to introduce another concept, and that is the concept of a modifier locus. A modifier is just a gene, but it's a gene that plays a really important theoretical role in evolutionary biology because it's a gene that influences how an organism lives and reproduces, how its DNA is passaged from one generation to the next. So in this body of theory, we're particularly focused on modifier genes that alter whether an organism reproduces mitotically or meiotically. And that would be a modifier of sex, or a modifier of recombination might influence the number of sites within a chromosome that undergo recombination, or the patterning of recombination across the chromosomes, whether they tend to be telomeric or well distributed across the chromosome. That would be a modifier of recombination. So we can track how a feature like sex would change within a population by writing down what happens to all of the combinations of genotypes in a population, including not just selected loci like this A locus here, but also modifier loci that alter the passage over time through sexual versus asexual routes. And I've only shown 2 of the many equations that would be had for this model. But what we basically do is write down the equations that incorporate selection, meiosis, or mitotic replication, and then repeat those equations over and over and over again to determine when the new modifier alleles can spread. But the real challenge for selective interference is that this is not enough. We have to also incorporate stochasticity. We have to include in these models not just how these equations change in an infinite population, but we have to account for drift. And that's challenging because these equations, we already have dozens of equations that are nonlinear, and now we have to account for stochasticity in those equations. But we've made some analytical progress on this front too. My colleagues and I have been using a variety of approaches, including branching processes to understand how sex and recombination affect the probability of fixation of a mutation. And by increasing the fixation rate of beneficial mutations, releasing them from that selective interference, the modifiers hitchhike alongside in frequency and rise and increase the amount of sex and recombination in the population. In other approaches, we've used moment methods-based methods to show how negative linkage disequilibrium develops in the presence of finite sampling and drift within populations, and how that comes to impede adaptation. So just as an example here, as one graph from one of these papers looking at the analytical predictions and curves and the simulation results. So we can make some headway into these tricky stochastic questions, making a wide variety of assumptions. When I started on this subject, I imagined that drift and stochasticity would play a really large role in small populations, but I had a hard time imagining that it would be that important in very large populations that we see for many species in nature. But I've come to appreciate that I was just plain wrong. And that is partly something that we can see from the molar diagram that Crow and Kimura drew. In larger populations, we're going to see more variation across the genome segregating at any one point in time. And with that increased variation, we're going to see more interference. There's going to be more neighboring loci with selectively impactful alleles than if the populations were small. Furthermore, if there are more loci, if there's weaker selection per locus, and if there's spatial structure so that drift happens within your local deme, then even in large populations, even in infinitely large populations we showed in this last study, then selective interference can be important and drive the evolution of sex and recombination. The other important feature that I'd like to talk about from this work is that really selective interference will act regardless of the form of selection. I've been talking a lot about beneficial mutations, but it works as well with deleterious mutations. Muller's ratchet being one example, but even in a population that is sexual, We showed that the amount of recombination, or the map length in Morgan's, would evolve to be higher in the presence of recurrent deleterious mutations due to this selective interference effect. Host-parasite interactions, so-called red queen interactions, also work, or combinations of these selective forces, beneficial and deleterious, beneficial and red queen, or deleterious and red queen. The key element is that with stochasticity, selection doesn't act on each locus independently. It acts on the cohorts of alleles that are found in the genomes present within a population. And after a period of time with drift and with selection, the combinations of chromosomes that remain tend to have a lot of this hidden variation, good alleles on bad genetic backgrounds. Modifiers that increase the rate of sex or recombination then hitchhike alongside the favorable alleles that are freed from this interference, as sex and recombination shuffles about that hidden variation and reveals it to selection. And now I want to turn to the last of the three acts, and that's really where we are now and where we need to go. I think the major question that remains in this field is whether selective interference is common and strong enough to dominate other processes and to account for the maintenance of sex across so many species in the tree of life. And by other processes, there are a lot. Epistasis, the fitness interactions between loci, dominance, costs of sex, spatial heterogeneity, local adaptation, migration, temporal heterogeneity. So the net result of selection facing a population and whether selective interference is a major player is still, I think, an open question. Let me spend a few moments talking about one of those other processes and how it interacts with selective interference. So I want to talk about epistasis. For population geneticists, we define epistasis as a departure from independence in these fitness interactions. So if the fitness of a beneficial combination is a little bit higher, we say there's positive epistasis because good alleles on good genetic backgrounds increase fitness a little bit more than you'd expect. So population genetics theory has gone on to show that there will be an alignment will develop between the genetic associations found in a population and the amount and the sign of the epistasis between the loci. So fitness tends to increase if there's a beneficial, a positive epistasis, the fitness of this double beneficial chromosome will be even higher than expected, and it'll come to become overabundant in the population relative to the randomized case, which means that there will be positive disequilibrium. So the disequilibrium lines up with the amount of epistasis in a population. And that means that when recombination happens, it'll tend to break up the very associations that have been built by past selection. And that'll tend to reduce fitness immediately in the offspring. We call that a recombination load. And by and large, most of the time, we're going to see a reduced fitness in the offspring. Only in these kind of oddball zones where there's disequilibrium that doesn't match the amount of epistasis in the population do we see the recombination load reverse and actually create an advantage to breaking apart the genetic associations that are present. There's a second zone I want to talk about, and that's where there's negative linkage disequilibrium. This is the hidden variation zone where negative genetic associations impede selection, good alleles hidden inside bad genetic backgrounds. And in this zone, there's an advantage to sex and recombination because it can reveal that hidden variation, increasing the amount of genetic variance in fitness and increasing the response to selection. So the combination models can show us where the net effect of all of those forces favors modifiers that increase sex and recombination, and that happens in these purple zones. The exact boundary of which depends on the parameters used. In an infinite population, where there's only epistasis and not drift, then it's in this region where sex and recombination are favored, and that region requires weak and negative epistasis. What does drift do? Well, if we can think about it on this very same diagram, drift, as I've mentioned, alongside selection, creates interference and more negative genetic associations. So the disequilibrium line moves down. And that, in turn, causes a larger region in which we see sex and recombination favored. Here is an example of simulations that drive home that point. On the x-axis is the population size. And so let's start here in an infinite population. What would we see?
Sarah?
Yes?
1 more minute.
Thank you. We see that negative epistasis is necessary in an infinite population, but in a finite population, the epistasis can be negative or positive. So rather than epistasis dominating, we see it's almost the reverse, that selective interference and these drift effects seem to be more important across a wide range of population sizes. So what evidence do we have for selective interference driving the evolution of sex and recombination? Few studies have actually studied this question in a manner that teases apart the mechanisms. And that's why I was so pleased to see the sorts of studies coming out of Anil Agrawal and Spex's labs that have examined species that undergo sex to see, does sex go up or down in frequency within a population? We can watch evolution in action and track how sex changes in these experiments. And they were able to show that we were in this region of the plot that I've been showing, which is consistent with selective interference, but not proof. But at least this type of experimental approach is promising, and I think in the future will help us to determine how important selective interference is in driving the evolution of sex. So I think we are still in an act in progress in understanding selective interference and its role in driving sex and recombination to evolve. Certainly, we know that selection in the presence of drift causes the accumulation of good alleles on bad genetic backgrounds, and this will limit the response to selection. That within a single population, sex and recombination can evolve and be maintained due to this selective interference. And I think this is probably the major force preventing sex from being lost or recombination rates from getting too low within species. Yet we still do not know whether selective interference is common enough empirically and whether relieving this interference is the main explanation for the ubiquity of sex. And with that, I'd like to thank my collaborators for the many excellent discussions and projects that I've been involved in. Thank you.
All right, thank you. So for those of you who have questions, you can push the question and answer button and formulate questions, and Sarah will be glad to reply to your questions. We're now going to move on to Abby Durenberg from UC Berkeley. We'll be talking about spatial patterning of meiotic recombination. Abby, it's yours.
You're still muted.
Those of you who have your bingo cards just got 1 square. It's a great pleasure to be here, wherever here is for all of you. I wish I could see you. And I really want to shout out to Anne-Marie Mahoney and Tracy Pellegrin and the rest of the GSA staff and everyone who participated in making this happen and so successful. And it's an honor to be here, to have the opportunity to talk about some of the work from my lab that has led us into an area I never really intended to explore, which is how meiotic recombination is patterned along chromosomes. And Sarah just reminded you that meiosis and meiotic recombination in particular underlie the transmission of genetic information in all eukaryotes. And yet there are fundamental mysteries about how it happens that we're still trying to address. So I'll remind you about the mechanics of meiosis as it happens in most eukaryotes. The goal of meiosis is to separate homologous chromosomes. So this is just a schema showing a nucleus with 2 pairs of chromosomes. The maternal and paternal copies will eventually need to separate from each other. And for that to happen, the chromosomes undergo a series of events. They have to somehow find each other in the nucleus. And as they do that, they undergo double-strand breaks. And they also undergo synapsis, which is what I'll talk about today. They zipper up through the assembly of a unique protein complex called the synaptonemal complex that was discovered in the mid-1950s. And in the context of this paired and synapsed configuration, the chromosomes can then undergo recombination. A subset of those recombination events lead to the formation of these structures called chiasmata, which physically link homologous chromosomes and enable them to segregate from each other by maintaining that contact until the first meiotic spindle is assembled. And then chromosomes release cohesion in a stepwise fashion to enable first one round of segregation to separate homologous chromosomes, and then a separate round to segregate sister chromatids. So I think it's really helpful to take a historical vantage point on meiosis by looking at this review or opinion piece that was published by the late great Robin Holliday, for whom the Holliday Junction is named, in 1977. So Holliday was, of course, very interested in meiotic recombination. And in this essay, he pointed out that there were three fundamental mysteries that still remained about meiosis. One of which was how chromosomes actually recognize each other to pair up. This is still a major mystery and something we're very interested in, but it's not the topic of my talk today. Another mystery was how meiotic recombination was initiated by the formation of breaks. And now we know quite a bit about that, but I'm not going to talk about that today either. Instead, I'm going to focus on this issue that was sort of a subtext of Sarah's talk, which is how crossovers are regulated to ensure that there's one per chromosome pair at the least, and yet the number per genome is much less than would be required to attain one per chromosome on a random basis. So my lab studies meiosis primarily in the nematode C. elegans, which is fantastic for a number of reasons. One thing that we really like is that you can see the whole process of meiosis because it happens in these two arms of the gonad, which produce both sperm and oocytes. And as cells go through meiosis, they progress through this tissue so that an adult animal has all of the stages of meiosis present in a snapshot. So hermaphrodites produce first sperm and then oocytes, which are fertilized as they pass through the spermatheca and then divide internally. So we can visualize a lot of aspects of meiosis in this system. And I was drawn to meiosis initially because I'm very interested in chromosome structure, and there's so many interesting things that happen to chromosomes during meiosis that we still don't understand. One of them is that chromosomes at the beginning of meiosis undergo a major structural rearrangement. So they replicate in an S phase, and then through magic that we are just starting to understand, the cohesin complexes, which we know play a major role in structuring all chromosomes, they undergo some kind of change in their activity that leads to the formation of an axis, a linear scaffold made of cohesins, from which the chromatin emanates as loops along a highly elongated conformation. And this morphological rearrangement is essential for everything else that happens. If it doesn't happen, there are no double-strand breaks, the chromosomes are unable to find their partners and pair, and they never synapse. In fact, the axis, which is shown in red here, is, we think, the substrate for the assembly of the synaptonemal complex between the chromosomes. And I'm going to try not to burden you with specific proteins more than necessary, but I'll just mention that the axis, as I said, has at its core cohesins, and then a family of hormodomain proteins that play essential roles in meiotic checkpoints and regulation. And the synaptonemal complex is made of a set of proteins, of which 6 have been identified in C. elegans. They are not well conserved. What they share is large regions of potential coiled coil formation. So that when the synaptonemal complex assembles between chromosomes, they take on this kind of very apparent paired configuration, but you can see that they're actually held at a distance. And this is somewhat strange because the whole point of pairing and synapsing chromosomes is that this enables them to undergo recombination, and yet they're actually held at this distance, and the space between them is where the synaptonemal complex is. And you can see that by electron microscopy. Here, this is a C. elegans synaptonemal complex, but it looks very much like SCs from other organisms. The structure and the dimensions of the SC are widely conserved among eukaryotes. It's usually about 100 nanometers in diameter, and it always has this striated appearance that have led people to describe it as a series of transverse filaments between the paired axes. Here, the dark stuff is the chromatin. And again, I mentioned that the proteins that make up this material are not well conserved across eukaryotes, but they all have extensive regions of potential coil formation. And we've known for many years that the SC is required for this phenomenon called crossover interference. It's not strictly required for crossovers in all species, but it is required for the crossovers that show interference. And so the work that I'm talking about today really started several years ago with the efforts of a former postdoc in my lab, Ofer Rogue, who is now at the University of Utah, and he was interested in studying the assembly of the SC between chromosomes, and he wanted to take advantage of the fact that C. elegans are transparent, and we can now label proteins in the germline, and he wanted to image this process of pairing and synapsis to study how the SC nucleated and spread out along chromosomes. And to do that, he successfully tagged one of the SC proteins, CYP3, with a GFP tag. And he worked out methods that would enable him to image living worms over a period of time so that he could follow this process of assembly from early meiotic stages into full pachytene when the chromosomes are fully paired and synapsed. And it took him a while to achieve this kind of long-term recording because it turns out that worms do not really like being put under a cover slip and immobilized for extended periods. But we worked out conditions under which he could actually image this process over more than an hour. But before we got to that point, he made a lot of movies where the worms would undergo this interesting metabolic arrest, which we eventually realized was due to the fact that they cannot eat under a cover slip. And we know from work in many labs that withdrawal from food triggers a whole bunch of physiological changes in C. elegans that can be suppressed by addition of serotonin. And so that's ultimately how Ofer was able to make these long-term recordings. But before we learned that we should add serotonin to the medium under the cover slip, Ofer made a lot of recordings that looked kind of like this next movie that I'll show you, in which the worms underwent this arrest. And we saw this weird phenomenon that we didn't really understand, in which the SC proteins, which were soluble in the nucleoplasm, would suddenly start to form aggregates in the nuclei. And of course, we were heavily influenced by a lot of papers coming out in various areas of cell biology that were showing that a number of different kinds of bodies inside cells assemble through a process of regulated condensation. And so we started to wonder if this phenomenon that we were seeing under this weird arrest conditions might represent condensation of synaptonemal complex proteins. And we decided to explore that further by testing whether the synaptonemal complex might be sensitive to hexane diol. Hexane diol is an aliphatic alcohol that when added to cells seems to reduce internal hydrophobic interactions. And many phase-separated bodies within cells seem to be sensitive to hexane diol. And so in this case, Ofer took a strain in which the chromosome axis was labeled with one fluorescent protein and the SC was labeled with another. And he exposed the tissue to hexane diol and immediately saw that the synaptonemal complex was very susceptible to dissolution by hexane diol, suggesting that it is held together by weak, possibly hydrophobic molecular bonds. So that was an intriguing finding. He also found, as is often the case in phase-separated systems, that this dissolution by hexane diol is reversible. So if you add hexane diol to C. elegans gonads, the SC comes apart, and then if you dilute the hexane diol, those proteins reassemble between the chromosomes very rapidly in the space of a minute or two, much faster than they normally assemble along chromosomes. So this started to smell like the synaptonemal complex might be assembling through a condensation mechanism. And so we wanted to explore this further. And one way that we did this was to look at what happens when the synaptonemal complex cannot assemble between chromosomes. So it's been known, again, since the mid-20th century that under a variety of conditions, the synaptonemal complex proteins, instead of assembling between chromosomes like they normally do, will form nuclear or cytoplasmic bodies that are many layers in which each layer appears to be a synaptonemal complex. And because of that, they were termed polycomplexes in this early paper by Roth. And and we thought that we could observe polycomplexes in C. elegans because it had been reported by Monique Zetke's lab that the absence of this key chromosome axis protein, HTP3, led to the appearance of these bright dots in each meiotic nucleus that were positive for synaptonemal complex proteins. And so we looked at these structures by electron microscopy, and we observed that they are indeed polycomplexes. They appear to be multimers of synaptonemal complexes. They recapitulate the precise spacing and this kind of striated appearance of synaptonemal complexes. So they're aggregates of SC proteins, but they are not associated with chromosomes. And so we could watch them behaving in cells and observe their dynamics. So we observed that these nuclear bodies, like synaptonemal complexes between chromosomes, are sensitive to hexane diol. Whoops, sorry about that. And so if we add hexane diol, they blow apart. But as the hexane diol is diluted, they reassemble and start to fuse together to form larger bodies. And we could also watch them in living worms not treated with hexane diol. In this case, the nuclei are marked by the fluorescence of this histone M cherry fusion. And what you'll see as I play this movie, hopefully, is that these bodies have liquid-like behaviors. These two polycomplexes will bump into each other and fuse. And then this nucleus is about to undergo apoptosis, which is a frequent event in oocytes in C. elegans. And you'll see that this body just disappears as that cell is engulfed. Oops, I'm having some video issues. Sorry. So here we go. And these two are going to fuse and form one larger body. And then this one's going to go away. And so these are liquid-like behaviors. This kind of dynamic behavior of a body implies that the molecules within it, in this case, the synaptonemal complex proteins, are able to move and rearrange themselves freely, which is a liquid-like behavior. And we wanted to test whether the proteins within SCs between chromosomes also show this kind of dynamic behavior. And to do this, Ofer tagged, again, one of the synaptonemal complex proteins with a photoconvertible fluorescent protein, mMAPLE. And he observed that if he was able to photoconvert a small portion of a nucleus from green to red, those red converted SC proteins would spread out through the SCs in that nucleus over a period of about 20 minutes. As a control, he also tagged one of the axis proteins, HIM3 in C. elegans, and found that it did not show this kind of dynamic behavior. It stays relatively put over a similar time frame. And so based on the observations that I've just shown you and others that I haven't had time to describe, we concluded and published that this synaptonemal complex is a biomolecular condensate, and specifically it's a liquid crystal. A liquid crystal is defined as a state of matter in which the composite molecules are free to move relative to each other, so that's the liquid part, but they have a defined organization. It can just be a preferred orientation, but in this case, they have a very specific order relative to each other and a structure that we can observe by a number of different microscopy techniques. So for the last several years, we've been thinking about how might this discovery that the SC has liquid-like behavior, how might that contribute to recombination patterning? And what do I mean by that? So I need to give you a little bit more background before I get into the specifics of this. So as I mentioned, we know that during meiosis, there are numerous double-strand breaks along each chromosome. Sometimes those breaks are necessary for the assembly of the SC, but sometimes not. And what it looks like in C. elegans is that all of the breaks initiate a process of homologous recombination. And you can see small foci along the chromosomes that are positive for a number of different meiotic recombination factors. And at some point in mid-prophase in C. elegans, one of those sites, always one, is selected to become a crossover. And what we see is that the recombination factors, the crossover factors, accumulate at that site and they disappear from the other sites. And in many organisms, there can be more than one crossover per chromosome, but as first observed by Muller and Drosophila, those are always widely spaced, and that's the phenomenon of crossover interference. Now, the role of the SC in crossover interference has been controversial, but there has been good evidence in C. elegans from Anne Villeneuve's lab that the SC plays an important role in this kind of crossover patterning. I also need to point out other work from the Villeneuve lab. They identified a protein that's required for crossovers. It's a cyclin homolog, and it's a beautiful marker for these sites along each chromosome where a crossover is specified. So there's 6 pairs of chromosomes in C. elegans, and at this late stage of meiotic prophase, we can observe 6 brightly stained dots of this COSA1 protein along in each nucleus, and those are the sites where the 6 crossovers will occur. They also showed that knockdown of synaptonemal complex protein expression is sufficient to partially abrogate crossover interference. So if you RNAi some of the CYP proteins that make up the SE, sometimes you get an increase in the number of crossovers along chromosomes. And we also found unintentionally, by making a tagged allele of one of these synaptonemal complex proteins, we found this allele that also seems to break crossover interference fairly seriously. So instead of the 6 designated crossovers we normally see, there are 12 to 13 on average in these nuclei with the mutated CYP4. So it seems the SC plays a role in interference, but we don't know exactly how. And so we speculated that there could be proteins that are somehow moving within this liquid-like environment of the SC that are somehow patterning crossovers. And our attention was focused on this family of proteins. In C. elegans, they're called ZHPs because they're homologous to ZIP3 in Cerevisiae, which was the founding member of this protein family. These are proteins that have a ring domain and a coiled coil domain and an unstructured C-terminal domain. And as I said, in C. elegans, there are 4 of them, and we found that they form 2 heterodimeric complexes. ZHP1 and 2 form a pair that interact, and they're required. Each of them is required for all of the activities of the other. Likewise, ZHP3 and 4 interact and are required for each other's localization and function. And what was intriguing to us is that all of these proteins localize to synaptonemal complexes and to these polycomplexes that form in the absence of an axis. That they are members of a conserved protein family. As I said, ZIP3 was the founding member, but these proteins appear to be present in all organisms that do meiosis, and they play an important role in making and patterning crossovers, but it's still not understood how they work. If we take away ZHP3 or 4, we do not see any evidence of recombination intermediates. We don't see these late COSA1 foci that I pointed out earlier, and we don't even see the faint foci that mark the early recombination events. Conversely, if we take away ZHP1 or 2, instead of getting 6 designated crossovers in each nucleus, what we see is a gradual accumulation of these very faint early recombination foci to a much higher than normal number, but these do not mature to become crossovers. So all of these proteins are necessary for crossovers and their regulation. And what we observed in both fixed and live images is that these proteins accumulate from the SC, so 2 of them, ZHP3 and 4, accumulate from the SC to the site of crossing over. And that's really obvious in this mutant DSB2, which limits the number of double-strand breaks so that there's often fewer than 6 crossovers per nucleus. And so you can observe an individual synaptonemal complex here in which the ZHP3 protein has moved to the one spot that's going to become the crossover, but it hasn't happened along any of the other chromosomes, presumably because they didn't have double-strand breaks. So we speculated that perhaps ZHP3 and 4 somehow form a pattern by accumulating at a subset of recombination intermediates to designate these widely spaced crossovers. ZHP1 and 2 do not accumulate at these late intermediates. They stay distributed throughout the SC, but they're required for this accumulation, and we don't yet know why. I mentioned that COSA1, the cyclin-like protein, is required for crossing over, and it localizes to these late foci where the crossovers will happen. And we wanted to identify whether it was working with a CDK. And we found that indeed that it works with C. elegans CDK2, which is expressed late in meiosis, and it goes to these foci that are exactly the same spots where COSA1 is. And we also know these proteins physically interact. So we now know that COSA1 and CDK2 are required not just for crossing over, they are required for crossing over, so you don't see any of these foci if you don't have COSA1, or in this case, we made an analog sensitive allele of CDK2, and if we treat with an inhibitor that inhibits its kinase activity, we don't get these late foci, and we see univalent chromosomes at dikinesis. But also, COSA1 and CDK2 are required even for the early stabilization of recombination intermediates, which we mark here with MUSH5, which is required for homologous recombination in meiosis. And so even those foci are missing. But we do not think that COSA1 and CDK2 or MUSH5 for that matter are diffusing within the synaptonemal complex. They all, COSA1, MUSH4, MUSH5, all of these proteins accumulate at these late recombination sites. Interestingly, ZHP3 and its partner ZHP4 remain dynamic at these sites. So if we, if we do a FRAP experiment where we bleach them, we see that they actually recover over time. But these other proteins seem to be locked at the site of the recombination intermediate, probably by binding directly to the DNA, and they don't recover if we do a FRAP, which I'm having trouble with right now. Okay. And this is just a quantification showing the recovery of ZHP3, but not of COSA1. And I haven't shown you any data for the other proteins, but just to give you a sense of how we look at these. So how is this all working? So those of you who've heard me talk about this in the recent past may have heard me talk about Turing patterns and other ways that recombination intermediates may form these widely spaced kind of nodes along the chromosome. But more recently, in talking to a physicist about our system, he actually suggested a new way of thinking about it that I think may be a better model for what we actually see and what we know about the mechanics of this system. This is the earliest description that I'm aware of recombination nodules, which were discovered by Adelaide Carpenter through serial section electron microscopy in Drosophila oocytes. And this is just her drawing of what a recombination nodule, so one of these late designated crossover sites, actually looks like along the chromosome. And you can see that it's actually a spherical body that's not within the synaptonemal complex, but sort of separate from it. And these are other images from tomato. These are actually early recombination nodules. And this is a more recent paper from Scott Hawley's lab showing recombination nodule. And you really get the sense that these are aggregates that are forming alongside, but not within the synaptonemal complex. Another hint was data from Ofer when he was in my lab. He found that ZHP3 is soluble in hexanediol when it's distributed along the synaptonemal complex, but when it accumulates in these late foci, it becomes resistant to hexanediol, indicating that it's undergone some kind of change in its chemical bonding that makes it more resistant to extraction. So all of this suggested that these recombination nodules are some kind of condensate that forms adjacent to, but not within the synaptonemal complex. And so what I'm going to suggest is that the way that these nodules may actually form along the SC is through a process of de-wetting, in which these proteins, the recombination proteins, particularly ZHP3 and 4, are associated with the SC, but they're not, they don't love the SC. And in response to a cell cycle signal that happens toward the end of meiotic prophase, they become less enamored of the SC, and so they beat up to form recombination nodules. And this is a well-studied phenomenon in fluid mechanics. People have studied the behavior of de-wetting systems of a wide variety of types. But one property of such systems is that as two materials that have limited affinity for each other separate and one de-wets from the other, the droplets undergo this process of coarsening or getting larger and fewer in number. And they do that either by colliding with each other or through a process of what's called Ostwald ripening, in which molecules, if they are free to leave one of the smaller droplets and travel into one of the larger droplets, will become more stable because there's less surface tension in the system. So what I'm suggesting is that these recombination nodules may form through a de-wetting process that eventually coarsens to produce a single large nodule per C-ligands chromosome. If the chromosome is long enough, you may actually get additional droplets at a distance. And I think this is really consistent with everything we see. For example, I didn't show you this earlier, but even these polycomplexes that are not associated with the chromosomes undergo what looks like a de-wetting process where the ZHP3 and 4 are distributed throughout these bodies early on. Then we see a dot of COSA1 and CDK2 on one edge. And eventually all the ZHP3 and 4 appear to sort of ball up and join that dot to form what looks like a recombination nodule, but there's no recombination in this mutant. There's not even any double strand breaks. And as far as we know, these are not associated with chromosomes. We think that the trigger that allows this dewetting or whatever this process is to happen is the shutting off of a key meiotic kinase, CHEK2. So CHEK2 is required for early events in meiosis, including double strand break formation. We know that it normally shuts off in response to satisfaction of what we call the crossover assurance checkpoint. And if we turn it off prematurely, we can actually see, sorry, that's what was shown here. We see precocious formation of crossovers in response to the loss of CDK activity. So we think that CDK2 and COSA1 are locally promoting the formation of these nodules by promoting aggregation of these recombination proteins, and that CHEK2 is acting globally to promote the association of some of these proteins with the SC, and then when it goes away, that's what triggers this process. So this is very speculative, and I definitely welcome your feedback. It's also consistent with the fact that we see this a structural change in the synaptonemal complex that accompanies crossover designation, which is consistent with the idea that the SC is actually an active participant in this process, and that some of the regulation may actually target the SC rather than the crossover proteins, or in addition to the crossover proteins. And finally, I want to point out one thing that I think is appealing about this model, which is that we know in some organisms that the formation of early recombination intermediates triggers formation of the SC. And this is easy to imagine as a reverse of the dewetting that I just described if these recombination nodules also can sort of wet the SC, if they can interact with SC proteins and thereby nucleate their assembly along chromosomes. And then later they will dewet from the SC to form these patterned recombination intermediates. I just want to point out that Robin Holliday actually suggested a really similar idea. Yes, I will stop for this process. So I will point you to this amazing paper that was extremely prescient in 1977. It described the SC as potentially a closed organelle in which proteins might diffuse and bind cooperatively. And I'll just thank my lab. I've highlighted work from Weston Stouffer and Liangyu Zhang, and some structural work from Simone Köhler, who's now at the EMBL in Heidelberg. Thanks so much. I wish I could take your questions, and I'll be happy to respond offline.
Yes, you can take questions, and people can formulate their questions online, and you're able to answer them whenever you want. So this is essentially concluding the TAGC, a portion of the TAGC, and I'd like to thank the three speakers who all gave really excellent talks. And I also would like to thank all the speakers of this big Genetic Society of American experiment. I would be tempted to conclude by saying that the experiment has been a great success, but the conference is not really over. There are all the poster sessions next week, and they're an integral part of the GSA conference. So they bring the whole community together and will now allow you to talk face-to-face and to exchange ideas and feedback through Zoom. It's important that we really maintain this tradition. And even though we are now virtual, you can use the app to view the posters and in some cases see short videos by presenters and interact with them. So starting Monday, you will be able to join Zoom meetings where you can ask questions of the poster presenters. And please take time to view some of the outstanding work on the posters and then turn up next week ready to ask a question or two for each of the poster presenters. We also have workshops that are typically associated with these meetings, the TAGC meetings, and they're also still ongoing. They're a bit delayed, but they will be starting on May 7th, and we'll be holding 1 to 2 workshops per week on Tuesdays and Thursdays. And again, you will need to register separately for each of these workshops in the week. So I'd like to thank a number of people because this has not been an easy enterprise. Being able to go from a meeting that was in person to a meeting online and doing this in a few weeks, I thought took a heroic effort of the Genetic Society of America. And so I'd like to obviously thank the conference organizers and the session chairs, especially the staff from GSA and the GSA board, as well as the Gruber Foundations, the sponsors and the exhibitors. There's a few vendors that have played a special role in this meeting, and none of this would really have been possible without the help of these nimble and dedicated vendors. And I have to specifically talk about Warp Speed, ATIV Software, and Fixture. I'm not familiar with them, but apparently the GSA people are, and they've provided a dedicated service. Finally, I'd like to thank you, the attendees watching right now from all over the world. For some of you, it's early in the night or late in the night or early in the morning. Just a few stats, because I think it's interesting to go through these stats. 371 people this week gave a talk. More than 9,000 people attended and viewed these sessions from almost 80 different countries. And there were more than 7,000 tweets and more than 5,000 Slack messages. So far, more than 4,600 hours of recordings have been watched. And you know you can watch many of these talks more. So I think it's been a very productive meeting. And even though I miss seeing all of you, I think this was really an interesting venture. So thank you for showing up for this conference and keeping our field moving along, even when most of us must stay at home. And so, you know, thank you for engaging with one another and TAGC. And we're hopeful that the collaborations that are sure to come as a result of this meeting will be productive. The global pandemic obviously is a clear demonstration of the importance of science, the importance of scientific community, and the importance of working together. And you should all be kind of enamored by the fact that there is a COVID outbreak. Finally, I think it's a good moment to remind all of you who have benefited from this TAGC meeting to show your support in becoming a member of the Genetic Society of America. You don't need to call yourself a geneticist or to be a GSA member. If you use genetic tools in your work and you're one of us. So you don't need to be an American to be a GSA member. We have members from more than 50 different countries, and you don't need to be a professor, an academic to be a GSA member. You can be a student, a member of a lab, a technician, or part of all sectors. So everyone is welcome and included in the society. So I want to make a special mention, finally, of Mark Johnstone, who has worked tirelessly for years on making this conference a success. And Mark has really been a leader and has brought us together on many occasions and has really worked tirelessly to make this a success. And I think we owe him some gratitude for that. So this time, I think it's time to finish and wrap up this section of the meeting. And I hope you'll all see posters next week. Thank you. All right. This ends the meeting, and you can all mute and leave. Thank you.